speaker, Dr. Jenny Sauk, who is my friend and colleague from UCLA. She's the director of clinical care for the UCLA Inflammatory Bowel Disease Center. And she is an outstanding clinician and colleague and speaker. So welcome, Jenny Sauk.
Hi, everyone. How are you? Hope you've had a lovely morning. It's really nice to be here in person finally, so it's really fun. Okay, so today actually what we're going to be doing is, my name is Jenny Sauk, as Dr. Israelian has mentioned, and I'm a gastroenterologist at UCLA, specializing in IBD. And I'm really lucky to be presenting today with Dr. Parambir Dulai. He's formerly a Southern California gastroenterologist. Now he's moved on to bigger things. He's a director of GI clinical trials and precision medicine at Northwestern, the Division of Gastroenterology at Northwestern. So we're lucky to present together. And I'd like to thank the organizers for this kind invitation. And today, what we're going to do is go over some of the more notable abstracts, IBD abstracts that we found this year. And just one note is that there's so many new therapies that are coming out down the pipeline. So a lot of our focus will be on some of the novel therapies that we'll be seeing in the near future. Okay, here are our disclosures. So before we get started, I'd like to just give a little bit of background of our treatment landscape for IBD. Over the last 20 years, we've seen such a major change in our treatment landscape. So before 1998 and the advent of Infliximab, our focus of treatment was really on symptom management. But after Infliximab was able to demonstrate mucosal healing, we've really seen a shift in our treatment goals, not only towards clinical remission, but also towards endoscopic remission. And with our novel therapies, novel technologies that we've had, we've really found new IBD therapeutic targets to look into. So we've had really an explosion of therapies over the last 20 years. And this includes not only biologics, but in addition to understanding some of the limitation with biologics, we also have new small molecule therapies that have been approved over the last couple of years. Even with all these novel therapies, though, I want you guys to all keep in mind that we're encountering a therapeutic ceiling. So for Crohn's disease, we're seeing that overall remission rates in our large clinical trials really are not reaching above 36% by week 50, and with 50 to 54. This is in Crohn's disease. And in ulcerative colitis, we're seeing the same trend as well. We're seeing treatment effect sizes of roughly anywhere from 7% all the way to 28%. And this is even with our more novel therapies like Ozanimod that was recently approved. So even when we take patients who are most optimized, so these are treatment naive patients who are started early on with combination therapy with Infliximab and azathioprine, mucosal healing rates in Crohn's disease are roughly 44%. And in ulcerative colitis, we're at 63%. So we certainly have more room for improvement here. So how do we break this therapeutic ceiling? I think this is something that we have to keep in mind. We have all these new therapies that are coming into the pipeline, but we also have to think about the big picture of what we can do to try to really optimize treatment effect. So not only are we going to be having novel therapies to be thinking about as one step, we have to really be thinking about earlier diagnosis, better risk stratification, really improved target-based management. And maybe we do need to be thinking about different therapeutic strategies or improving our positioning of therapies. But all of this has to be happening concomitantly in order for us to really achieve higher population level remission rates that we want to achieve. So some of the abstracts that we are going to talk about today will address this therapeutic plateau and how we're going to overcome that. So Dr. Dulai will focus on some of the novel mechanisms of action for therapies. We'll see, do we need to be considering longer induction periods for our IBD patients? Can we optimize drug levels, or can we do something to maximize the therapeutic effect of our current biologics? Should we be thinking about combination biologic therapies? So there have been a lot of emphasis of that with that in IDDW this year. So we'll be looking into some of these abstracts. And then lastly, I think it's important to, there were some really good abstracts looking at treatment withdrawal. So we'll be talking about that, since that's one of the most frequently asked questions that I think all of us get in this room. And lastly, we'll touch upon IBD therapeutics in the elderly. So with that, I'm going to turn it over to Dr. Dulai, and he'll talk about novel mechanisms of action for IBD.
All right, thank you everybody, and thank you for the warm introduction. It's great to be back, a little bit warmer here usually than it is in Chicago, but Chicago is actually quite nice this time of the year. So I am tasked with talking about novel mechanisms of action. I'm going to start by saying that the landscape of inflammatory bowel disease is beginning to dramatically shift, and we're seeing a huge influx of therapeutic mechanisms and novel mechanisms. So what I want you to try to take away from this is that there's sort of refinement in the way that we're thinking about therapies and targeting these diseases, and we are seeing some incremental benefit in terms of effectiveness and efficacy, and how we think about positioning some of those is going to become really important over time. So when we think about, okay, there we go. So one of the first mechanisms that I'm going to talk to you about is the IL-23 inhibitors and the P19 antibodies to the development for both ulcerative colitis and Crohn's disease. And so some of you are, familiar with this mechanism because of Ustekinumab or Stelara, which targets both IL-12 and 23, because it targets the P40 subunit that you can see here on the figure. The newer mechanisms are targeting that purple P19 under the IL-23 mechanism. So it has a bit more specific IL-23 inhibition with no IL-12 inhibition. And that is thought to be, or has been thought to be, sort of more beneficial because the IL-12 pathway does have some important anti-inflammatory properties that might be knocked off. And so having specific IL-23 inhibition has been thought to be more efficacious, and psoriasis has actually been shown to be more efficacious. So when it comes to IBD, there's 4 therapies in the pipeline. There's Risankizumab, Guselkumab, Brazikumab, and Mirikizumab. And I'm going to talk through some of the DDW abstracts that highlight some of the efficacy that we're seeing for these therapies in our clinical trials now. So before we get into the DDW abstract, throughout this, I'm going to give you some framework of what we've been published for some of these things. So this is the induction data in ulcerative colitis for the known available IL-12/23 inhibitor Ustekinumab, and then the soon to be available Mirikizumab, which is an IL-23 inhibitor. So what you can see from the Unify study on the left is that the 8-week outcomes for clinical remission, which is complete resolution of rectal bleeding and improvement in stool frequency, and clinical response, which is improvement in those, but not achieving clinical remission, was significantly higher for the Ustekinumab treated patients compared to placebo. And you can see the delta there for the clinical remission in dark green is about 10%, and the delta there for clinical response is about 30%. So those are the deltas you're looking at for Ustekinumab for an 8-week induction. Mirikizumab, when you look at the deltas there for clinical remission, you're talking about 22% in the dark green versus 5% in the placebo. You're talking about an over 15% delta, which just at face value gives you some opportunity to think about this therapy. And then clinical response rates are, again, almost a 40% delta difference. So the efficacy seems to be a bit higher, and we're very excited by this therapy. And we were sort of very excited about this novel mechanism. What was presented at DDW was the phase 3 trial, which has led to the FDA approval for this therapy. So Mirikizumab is a humanized IgG4 monoclonal antibody against the P19, which we talked about. Lucent-1 was the phase 3 induction study, which showed superiority of Mirikizumab, and that was presented at ECHO this past year, where they showed superiority for induction of clinical remission and other clinical, endoscopic, and histologic outcomes. And this is given as an infusion every 4 weeks for 12 weeks for the induction study. What we're going to talk about is some of the maintenance data that was presented at DDW, looking at patients who had responded to induction, who were re-randomized to Mirikizumab or placebo, and how the efficacy looked like when you started talking about maintenance therapy. So this graph is a summary of the maintenance data at week 52 for Mirikizumab for achievement of clinical remission, which is resolution of rectal bleeding and stool frequency scores, and the achievement of endoscopic improvement, steroid-free clinical remission, endoscopic remission, which was Mayo 0 or 1, which is basically the absence of friability, bleeding, ulcers, and then the evolving endpoint of histoendoscopic mucosal remission, which is both histologic and endoscopic remission. And I want to give the company credit because they actually added a new endpoint for this trial, which was bowel urgency. So in routine practice, one of the most common complaints we get from ulcerative colitis patients is my urgency is limiting me from being able to go out. I need to be close to a bathroom. And so they actually created a new bowel urgency score and looked at bowel urgency remission. And you can see here the efficacy rates for maintenance are in the 50 to 60% range for the mirikizumab treated group in blue, and the placebo rates were significantly lower in the green bar. So there was significant, very significant differences for maintenance data in terms of efficacy across every single endpoint that we would care and patients care about, which is, I think, another big part about some of these emerging therapies. They took really patient-oriented symptoms in some of these trials. So when we then shift focus and start talking about Crohn's disease, I want to give you a little bit of framework for the background around some of the data that's available for several therapies currently available. So on the left, you can see the Ustekinumab data. So this is the Unity 1, Unity 2 data, which led to the FDA approval for this therapy. And you can see, again, clinical remission, this time defined by CDAI, and clinical response. You have a delta of about 13% for clinical remission in Unity 1, and you have a delta of about 20% for clinical remission, the dark green versus the solid dark gray for placebo. When you start looking at the selective IL-23 inhibitors, brazikumab, risankizumab, guselkumab, and mirikizumab, when you start looking at the delta differences here, you can see there's a quite striking difference in terms of delta differences for induction data. And I'm just going to call your attention to guselkumab and mirikizumab at the bottom, where for guselkumab, you're looking at 54% remission rates for induction compared to 15%. That is a massive delta that nobody really expected for this therapy for Crohn's disease for an induction trial. So you're talking about 12 weeks, which is not typical because most of our Crohn's data, really you start to see the benefit at about 6 months. The induction data for guselkumab was sort of published recently, just sort of within the past month. And so this was, just to give you some framework here, so what they did was it's sort of an innovative approach where they did an induction trial and compared it to placebo, but they also had the comparator arm of ustekinumab. So the goal here for them was to compare different doses of guselkumab, 200, 600, and 1200 milligrams, against placebo, which they would need for FDA approval purposes. But they also went one step further and actually compared guselkumab to ustekinumab as a reference arm to give us a sense of whether there was a signal of potential superiority for IL-23 inhibitors against IL-12/23 inhibitors. And on the graph on the right, you can see very obviously that every dosing strategy for guselkumab was superior to placebo in gray. So you can see the different colors of teal. But what I want to call to your attention is that purple box on the far right. So that's the data for ustekinumab in terms of change in their symptom activity during the induction period. And you're seeing some slight differences there early on for the induction data, especially for the higher dose. So what was presented at DDW was the maintenance data. So what they did was they then took a maintenance study where they compared just guselkumab to ustekinumab to try to understand what the comparability or possibly the superiority was. I will caveat that by saying the trial was not powered to assess superiority, and this was really just sort of a bit more of an exploratory, confirmatory understanding of what was going on. So when we look at the maintenance efficacy data for clinical remission, a CDAI of less than 150, steroid-free clinical remission, endoscopic response, now we're talking about endoscopic outcomes, which was an improvement in SES-CD, and clinical remission plus biomarker remission, a CRP of less than 3 or a fecal calprotectin of less than 250. The green bar is the ustekinumab efficacy at week 48, and you're looking then at different doses of guselkumab, 200 IV versus with 100 sub-Q maintenance, 600 IV with a 200 sub-Q maintenance, or 1200 IV with a 200 sub-Q maintenance. And you can see here, when you look at clinical remission rates, the 600-200 group had 70% rates for clinical remission at 48 weeks. And numerically, this is about a 10% delta difference between ustekinumab. It's not intended to draw comparisons, but you can't help but draw a comparison for that superiority there. What I want to call to your attention is the endoscopic response rates. And you can see here that the guselkumab had almost a 15% higher rate of endoscopic response compared to ustekinumab. That's a pretty striking difference for two active biologic therapies to show in terms of numeric differences at 1 year. And so I think they're starting to be able to sort of confirm and will confirm with future phase 3 trials that this IL-23 selective pathway might actually be superior to what's currently available, which is the IL-12/23 inhibitor in ustekinumab. Shifting gears a little bit, we're going to talk about a newer concept or mechanism that we're really unaware of in the immune-mediated inflammatory disease conditions as they relate to GI. They're the S1 receptor modulators. So in order to understand S1P sort of agonists, I think it's important to understand the S1P mechanism. So when a lymphocyte tries to leave a lymph node to go to a site of inflammation, it follows this sort of trail of blood that's left, which is the S1P receptors. It's like sharks following a trail of blood. And that causes them, that gradient causes them to migrate from the peripheral lymph nodes to the central site of inflammation and to enter that tissue and help deal with the localized site of inflammation. So that S1P gradient that we're talking about is sort of takes it from the lymph node, migrates to sites of inflammation, then allows it to enter the tissue. And there's a bunch of different tissues that we're going to go through. The goal with S1P receptor agonists is to activate this in the lymph nodes and essentially trap them into the lymph nodes, a bit of a Hotel California type of situation. And if they can't leave, then they can't go to the sites of inflammation and contribute to the process of inflammation in chronic immune-mediated inflammatory conditions. So, under physiological conditions, only about 2% of lymph nodes are actually in circulation, but during times of activate inflammation, this S1P mechanism ends up driving these immune cells to leave and get into the peripheral circulation. So the target is to activate the S1P receptor in the lymph nodes to prevent them from getting out, but there's different tissue systems that these different receptors are located, and this is really important as we think about the S1P mechanism in practice. So you have brain, lung, lymph nodes, heart, and kidney. The S1P receptors are located really throughout the body, but different receptors are located in different places. The three therapies that are currently sort of either being studied or available are fingolimod, which has a non-selective S1P1 to S1P5 receptor modulator. So it's going to target every tissue. That's really most relevant when it comes to us in terms of the heart. And this first dose bradycardia that can sometimes be seen because of the mechanism. Ozanimod, which you're going to see some data on now, is a selective S1P1 and S1P5 receptor modulator. So that has a bit more selectivity to the brain so you're thinking about MS patients, the lung, and then really the gut and the lymph nodes that are sort of dealing with the gut inflammation. And then Etrasimod, which is an S1P1, S1P4, and S1P5 receptor modulator, has some really interesting data that was presented here at DDW, which we'll also review. So I think this is important because this concept is new to our field, and it's important for you to think about how it works as you think about selecting some of these therapies once they're actually in practice. So Ozanimod, the one that is available right now for ulcerative colitis in routine practice, this is the prior data that led to its FDA approval that was published in New England Journal of Medicine. I think it's important to level set what we're going to talk about with the DDW abstracts. So what you can see here for clinical remission, so this is improvement in stool frequency, rectal bleeding scores of 0, and endoscopic improvement. The delta here is about 5% for clinical remission, which was not significant in patients who had prior anti-TNF exposure. So the left side is people who had prior anti-TNF exposure. So there really wasn't as much efficacy if you'd had a prior anti-TNF therapy, whereas clinical response was significant. When you go to the no prior anti-TNF group on your right, you can see that the clinical remission rates are looking at a delta of over 15%, which was highly significant, and clinical response rates were up in the 50% range, as well as endoscopic improvement and mucosal healing. So this mechanism seems to work best before an anti-TNF agent. And that becomes relevant for some of the positioning points that Jenny had made earlier. I want to call sort of to the attention as well that they had week 52 data for their maintenance data in that trial, and you're looking at clinical remission rates of 37% compared to 19%. So this therapy is on the market based on these sort of phase 3 data that were published. But what was presented at DDW was a little bit more of an update in terms of safety, durability, and the new mechanism the new drug that's come out with Etrasimod. So this is the long-term study for Ozanimod, or the long-term safety study that was presented. And it's important to understand their long-term safety study a little bit to understand sort of what it was. So like I talked about, so there was the induction study. So they had 2 cohorts that came in, and then responders could go on to either placebo, Ozanimod, or Ozanimod, just different ways that they were randomized. What we're going to talk about is the open label extension. There's 3 ways to get into this. You could be a non-responder after induction. So maybe you got placebo, maybe you got Ozanimod, you went in, you got Ozanimod, open label for the whole thing. Or you had a disease relapse during the maintenance phase, or you completed 52 weeks of maintenance and actually entered the open label extension study to look at 2-year outcomes. What was really important in this study is they actually had endoscopic data as well as other information that they collected. So what I'm going to look at, what they presented was sort of an all-comer for that population. One of the big things with some of these drugs that are coming out in this mechanism is they're called small molecules because they're not as dependent on clearance and they have a lot less of an issue with bioavailability. So one of the things we've always wondered is there enhanced durability? Because you won't lose response, you won't get immunogenicity, you won't get antibodies. And what you can see here is that the clinical response rates out to 2 years was 98%. So you're looking at the time of entry, and then out to 2 years, you're looking at 98% of them were able to maintain that response out to 2 years. Typically, maintenance of response out to 2 years is about 60%. They had a non-response imputation, which suggested it might have been closer to 55%, but the study's not done. So they had to exclude a bunch of patients who hadn't actually gotten through to open label. What was also improvement is they looked at endoscopic improvement and corticosteroid-free remission out to 2 years. You're talking about 70 to 80% rates for all of these outcomes. So this abstract and the durability data that we're going to get from this is really going to help us solidify that these small molecules in this new mechanism is both effective and durable. So when you start it, you have less concern about whether you're going to lose response over time for the patients who do well. And the other important thing here was that there were patients who got in here because they had a disease relapse, and 60% of those who lost response were actually recaptured when they went into the Ozanimod open label extension study. So there's an opportunity to, if they stop for some reason, you can restart it and actually do well with them afterwards. The other one that was published, which was a phase 3 study, was the Etrasimod data. So similar mechanism, slight differences in the receptor modulator, and I've sort of point some of that out. So it's a once daily S1P receptor 4/5 modulator. The Elevate UC12 was a 12-week induction study, and the Elevate UC52 was a treat straight through design. So they got 52 weeks of therapy and they looked at treatment efficacy. The only difference between this and the Ozanimod is the S1P4, which has some cell-specific autoimmune modulation, which we're not entirely sure whether that's of clinical sort of importance. So here's the induction and maintenance data. So what you're looking at here is clinical remission, and I'm going to sort of orient you here. So the green bar on the bottom is the placebo rates at 12 weeks. The light blue is Etrasimod at 12 weeks. So that's the induction comparison. The orange is placebo out to 1 year, and the blue is Etrasimod out to 1 year. So that's the maintenance data. And you're looking at highly significant differences for Etrasimod versus placebo at both induction and maintenance. And you're looking at overall rates for clinical remission and endoscopic improvement that are up near 30–40%. So still not quite breaking that therapeutic ceiling that we were hoping for in ulcerative colitis, but still a highly efficacious therapy that's going to be available hopefully in the near future for us to use along with some of the others that are available. So the last thing I'm going to end with is one of the things you may have noticed is that some of the induction studies had different time points for their assessment. So some had 8 weeks, some had 12 weeks. And one of these concepts that's emerged with these oral pills that we're getting is, do you give a higher dose for a longer period to get them into a better state of remission and then maintain on a little bit lower dose, which we saw with tofacitinib. One of the drugs that we're really excited about is Upadacitinib, which is a JAK1 inhibitor. It's a selective Janase kinase inhibitor, has been shown to be safe and effective at 45 milligrams for an 8-week induction for moderate to severe UC. So that data is published. They had significant improvements in UC symptoms as early as day 1. So you're talking about rapid improvements in rectal bleeding score within a day or two of starting therapy. And patients who achieved early symptomatic improvement were more likely to achieve clinical remission or response at week 8. So we know these patients, they start the therapy, it does really well quickly. They're going to do well at 8 weeks. What this study wanted to understand is, among the patients who were able to maintain a higher dose further out, does that higher dose benefit the other patients? The patients who may not have had that rapid improvement early on and achieve a better induction response if you give a bit of a higher dose further out. So the non-responders at 8 weeks who got either 15 milligrams or 30 milligrams of Upadacitinib. And so what you can see here is they had 125 patients who had failed to achieve response at 8 weeks. 48% of them achieved a clinical response by week 16 and were randomized to 30 or 15 milligrams. Among those who responded at by week 16, the 30 milligram dose of upadacitinib actually achieved higher rates of clinical remission, so 33% versus 19% at 1 year, higher maintenance of clinical response out to 1 year, so 66% versus 36%, and higher rates of endoscopic improvement. So what they really started to argue here is that for the patients who don't have that really rapid early response, once you get them out through the 16-week induction, to maintain them on a higher dose, and you get a little bit more of an improvement over time and maintenance of that response over time. So a little bit of opportunity for dose optimization in a proactive manner based on their clinical parameters that you're seeing. And then the other thing that was presented here was a shift from both ulcerative colitis to Crohn's disease. So we talked about the JAK1 inhibitor. Tofacitinib did not do well in Crohn's disease. It didn't meet any FDA approval criteria, was not taken through. And so tofacitinib, which is a pan inhibitor, is only available for ulcerative colitis. But upadacitinib, they presented the Crohn's data for induction in moderate to severe Crohn's disease. And this was a striking difference, and I think maybe a little bit surprising to see such a benefit, where they had significantly higher rates of clinical remission, steroid-free clinical remission, endoscopic response, which is improvement in SCSCD or improvement in ulcers and some of those things, and endoscopic remission. So you're talking about endoscopic remission again of 20% within 12 weeks of starting this therapy, compared to the available JAK inhibitor tofacitinib, which actually did not meet any of these criteria in the phase 2 studies. So in conclusion, for some of these novel therapies, there are novel IL-12, IL-23 inhibitors, which look really promising. The S1P pathway looks like a very safe, effective, and durable treatment option for our ulcerative colitis patients. And the JAK1 inhibitors, I think, are going to revolutionize the landscape of post-anti-TNF therapy, especially in both ulcerative colitis and Crohn's disease. And I think one of the really important things to take away from here is there's a lot of promise and potential with the new pipelines that are coming out, but there's still a ton of room for improvement, which Jenny's going to talk about with some of the other abstracts that were presented. So thank you.
Great. Thank you very much, Parambir. Okay, so we're going to go on. I'm going to talk a little bit about positioning of therapies. There was an abstract that I thought was really interesting in light of the data that Dr. Dulai has just mentioned, incorporating some of the newer data that are out available. So this was an abstract that looked at biologic drugs for induction of remission in Crohn's disease, and this was using Cochrane's systematic review and methodology. And this was looking at probably the most recent relevant data we have for induction of remission of Crohn's disease. It included 46 randomized controlled trials with 13,882 participants, and it investigated 18 different biologics, combinations, active comparators against placebo. And in this network meta-analysis, they basically found that 12 out of the different biologics that were looked at achieved significant efficacy over placebo. And what they did next was looked at relative rank order of efficacy. Now we have to take this for a grain of salt. Our best studies, when we look at positioning of treatments, are head-to-head clinical trials. These are expensive and there are not very many of these types of studies. So sometimes we can glean a little bit of positioning of therapy from looking at network meta-analyses. So there have been several others published. This one I thought was interesting because it included some of our more recent data. So here they found that infliximab azathioprine still ranked as the first in terms of relative efficacy. This is followed by the biosimilar CTP13 infliximab biosimilar with azathioprine. This is followed by infliximab and then followed by guselkumab. And I'm probably from that robust data that Dr. Dulai just mentioned, that this is why this is kind of ranked higher. This is next followed by filgotinib, which is a selective JAK1 inhibitor. And then you can see the rest of the drugs here in the rank order of efficacy. Now, this is, some of the randomized controlled trials were of moderate quality. And, we have to really see the population of patients may have been different. Some of them are treatment naive or not. So this is just a little bit of an idea of where these therapies should be positioned. But of course, we're going to be looking more from real world data. I thought this was interesting just to give us a sense of where these therapies may be positioned for us for Crohn's disease. Okay, so I'm going to move on and talk a little bit about optimization of therapeutic drug levels. So this is another way perhaps we can enhance our therapies that currently exist. So I thought this was an interesting study that looked to see if we can improve the pharmacokinetics of Ustekinumab and Vedolizumab with combination therapy. We already have data available that suggests that combination therapy with Infliximab, with with immunomodulator, whether that's thiopurines or with low-dose methotrexate, we already know that that can optimize the pharmacokinetics of infliximab based on previous studies. However, we don't know how combination therapy really affects the pharmacokinetics of vedolizumab and ustekinumab. So this was a nice prospective study, the combo IBD study, that aimed to assess the correlations between thiopurines, 6-thioguanine, and use of oral methotrexate with trough concentrations of infliximab, ustekinumab, and vedolizumab, and anti-drug antibodies, and looking at clinical and endoscopic outcomes. And in this study, it was a prospective cohort with enrollment between 2017 and 2021. And they looked at Crohn's disease and ulcerative colitis patients on maintenance therapy for over 30 weeks. And patients could be on monotherapy or combination therapy with either methotrexate, low dose, or thiopurines. And that primary outcome was drug levels, anti-drug antibodies for infliximab, vedolizumab, and ustekinumab, and secondary outcomes was steroid-free deep remission and endoscopic remission. So in total, they had 367 patients. Infliximab, they had 113, and you can see the breakdown below. With vedolizumab, 133 patients, and ustekinumab, 123 patients. And again, you can see the breakdown of the patients below. And what you can see here for infliximab, the median infliximab trough levels in this group was 12.3. And not surprisingly, on monotherapy, you're seeing really high levels of anti-drug antibodies at roughly 35%. And this is much lower in combination therapy groups with thiopurine or methotrexate. And then when you look at infliximab trough levels in mono versus combination therapy, again, not surprisingly, we're seeing higher trough levels with infliximab combination therapy. And it doesn't seem to matter whether you're on methotrexate versus thiopurines. Those both achieve that higher trough levels that we want versus monotherapy. And there's no statistical difference with whether you use the methotrexate versus thiopurine. And when we look at the thiopurine combination, really what's important is that we need to consider optimized thiopurine levels. So Dr. Yarer had already demonstrated this in another study, but here you can see again that you do need to have 6 TGN levels above 146 here in order to be able to achieve the trough concentrations that are statistically significantly higher than infliximab monotherapy. Now with infliximab combination therapy with methotrexate, you're also able to achieve that significant higher trough level compared to monotherapy. And then when you look at rates of steroid-free deep remission, there's statistically significantly higher rates of steroid-free deep remission here if you're on combination therapy. Endoscopic remission rates are also higher on combination, but this was not significantly different. So what about vedolizumab? So we see median vedolizumab levels of 8.9, and you can see as we kind of understand about vedolizumab that there's not high levels of antibodies that are seen here, but 1.2% of patients on combination therapy did develop anti-drug antibodies here. But what's interesting is when you look at the vedolizumab trough levels, you don't really see much of a difference whether they're on combination therapy or monotherapy. It stays pretty much plateau. So you don't see that higher level that we see with infliximab. And same thing, if you try to optimize the thiopurine, you don't really see higher levels of the trough levels for vedolizumab either. And then when you look at steroid-free deep remission and endoscopic remission rates, those are not significantly better whether you're on combination therapy versus monotherapy. So what about ustekinumab? Are we seeing the same thing? So we're seeing median levels of 5.8 and really not high levels of anti-drug antibodies, whether you're on combination or monotherapy. And again, when you look at those levels, when you're on monotherapy versus combination, those levels stay pretty steady. And once again, with the thiopurine levels, even if they're higher, you're not seeing higher levels, they stay relatively steady. So then when you look at long-term outcomes here, this is now beyond 24 to 48 weeks, rates of secondary non-response for the infliximab group are roughly 30%. And here they could predict factors associated with non-response, and that included infliximab levels less than 12.3, 6 TGN levels less than 146, or infliximab monotherapy. But really they could not identify factors associated with secondary non-response based on kind of pharmacokinetic data for vedolizumab or ustekinumab. So in this study, the combination therapy for infliximab and oral methotrexate or thiopurine with optimized 6 TGN really do improve the pharmacokinetics of infliximab, and oral methotrexate could be a good alternative for combination therapy. And this is the first prospective study to really show that combination therapy does not improve the pharmacokinetics of vedolizumab or ustekinumab. So I think that's important to kind of really solidify here. Okay, we're going to move on. So there are several other abstracts I thought were interesting that looked into combination biologics. So Dr. Dulai did a wonderful job of summarizing some of the novel therapies. Now, what happens if we start to bring them together? Can we break that therapeutic ceiling that I talked about earlier? Can we achieve higher clinical response and remission rates? So this was an interesting proof of concept study that combined guselkumab and golimumab in moderate to severe active ulcerative colitis. And this is results of week 12. So this is an interim analysis. They're looking eventually later at monotherapy patients. I'll explain that to you later. It's called the VEGA trial. And here, as Dr. Dulai mentioned, guselkumab is an IL-23 P19 subunit antagonist, and golimumab is a TNF-alpha antagonist. So preclinical studies in murine models of active colitis have suggested that if you have combination inhibition of IL-23 and TNF-alpha, perhaps there could be combined therapeutic benefit by having that combination inhibition, possibly with wound healing network. So the question here is, can combination therapy with guselkumab and golimumab increase treatment responses? So this was a phase 2A study, proof of concept study, to evaluate the safety and efficacy of combination therapy through week 12 of guselkumab with golimumab versus guselkumab monotherapy or golimumab monotherapy in moderate to severe ulcerative colitis. So patients were randomized from 1-to-1-to-1 fashion in these three groups that I just mentioned. And the primary endpoint was clinical response at week 12 with major secondary endpoint of clinical remission based on the full Mayo score at week 12. And what I mentioned about this monotherapy phase, this is just data for week 12. So what happens after this is that patients then would go on to a monotherapy phase where they go on to either golimumab or guselkumab monotherapy. And so patients that could be included in this study had moderate to severe ulcerative colitis. They had to be naive to TNF-alpha antagonists and have had an inadequate response to conventional therapies. They had to stop their immunosuppressants prior to randomization, and they can be on up to 20 milligrams of prednisone, but had to have a mandatory taper by week 6. So when you look at patient characteristics, they were relatively matched throughout the groups. And when you look at the primary endpoint of clinical response at week 12, you can actually see really high levels of clinical response here at 83.1% in the combination therapy group. This is compared to 74.6% in guselkumab monotherapy and 61.1% in the golimumab monotherapy group. And you can see a statistically significant difference in that combination therapy for clinical response versus golimumab monotherapy. Then when you look at clinical remission, this is based on the full Mayo score. Combination therapy was associated with 36.6% clinical remission. And this is compared to 21.1% in guselkumab and 22% in the golimumab group. But if you look based on the modified Mayo score, which removes that physician global assessment, we see higher rates of clinical remission of 46.5% in the combination group versus 24% in the guselkumab group and 25% in golimumab. So we're seeing significant differences in clinical remission at week 12. And when we look at endoscopic improvement, we're seeing the same trends. We're seeing higher percentages of endoscopic improvement with combination therapy, roughly 49% in combo versus 29% in guselkumab group and 25% in golimumab monotherapy group. And endoscopic normalization is where you actually see absolute no erythema, just complete normalization. It's about roughly double in terms of the endoscopic normalization rate here at 18% in the combination group versus 8% in guselkumab and 9.7% in the golimumab group. And Dr. Dulai mentioned this composite endpoint of histologic remission and endoscopic improvement. So this is something we're going to be seeing in a lot of clinical trials. That histologic remission is supposed to be absence of neutrophils from the mucosa, no crypt destruction, no erosions and ulcerations or granulation tissue, and also endoscopic improvement, which is that subscore of 0.1. So it's a very stringent endpoint here. And what you can see is that that combination group had a composite endpoint rate of 41% versus 26% in guselkumab and 15.3% in the golimumab group. And when you look at key safety findings through week 12, overall, you see that adverse event rates are pretty similar across the groups at 40% in the combo group, 48.8% versus 43.7% in guselkumab and 52.8% in golimumab. When we look at serious adverse events too, we're not seeing significant differences within the combination group versus the guselkumab or golimumab group. There was one case of influenza sepsis in one patient in the combo group, but the patient was able to restart therapy after week 6. So in conclusion with this study, this shows that combination induction treatment with guselkumab and golimumab had greater proportions of patients achieving the following endpoints at week 12 of clinical response: remission, endoscopic improvement and endoscopic normalization, and the composite endpoint of histologic remission and endoscopic improvement. And adverse event rates were comparable among treatment groups. So this was one of the combination therapy trials that we saw. There was one more that I just think deserve mention, and forgive my poor photography here. So this was triple combination therapy with vedolizumab, adalimumab, and methotrexate in patients with high-risk Crohn's disease. And this was an interim analysis from the open label phase 4 Explorer trial. And patients had to be relatively treatment naive. So they had to have no previous biologic therapy. Their diagnosis needed to be less than 24 months. They had no prior surgery and no fistula. So they enrolled 55 patients. There was no placebo arm, and patients received vedolizumab, adalimumab, and methotrexate. And methotrexate, they achieved a little bit longer than week 26 in case there was relapse in the monotherapy phase. And so this is the data at week 26, the primary endpoint. And the study is ongoing, and they're going to have the monotherapy phase where they have open label vedolizumab, and the final efficacy endpoint will be at week 120. And here, the primary endpoint was endoscopic remission at week 26, and the endoscopic remission rate was 35% in all enrollees. And 46 patients of the 55 completed the study. So the rates of observed cases, 42.2% of endoscopic remission, with clinical remission at week 26 of 54% and endoscopic healing at week 26, 43.6%. So the most common adverse events here reported were arthralgias, nasopharyngitis, gastroenteritis, but overall had a pretty good safety profile. So what we're seeing with these combination trials is that the combination therapy seems to be during the induction phase, and then they're transitioning over to a maintenance phase of monotherapy. So I think we're going to be seeing a bunch of these studies in the future. And this may be the future of our therapies, that we'll be combining a lot of therapies together to try to achieve higher clinical remission and response rates for our patients. So the key will be looking at safety data over the long term. Okay, so we're going to shift gears, and I'm going to talk a little bit about withdrawal of therapy. So how many of us have patients constantly asking about when they can stop their treatments? Okay, yeah, so you get them better, you work really hard, and then all of a sudden they're like, doc, please take me off my drugs. So yeah, so this is, I thought, a really nice study to really talk about the data. This is data you can share with your patients about the rate of relapse when patients withdraw therapy. So this is the SPARE trial. We've been anticipating this for a while. It's from the Jeted group from France. And basically, we know that combination therapy with infliximab and an immunosuppressant, this is standard of care for our treatment naive, moderate to severe Crohn's and ulcerative colitis patients. But once you're in an established remission, we don't really have a lot of data to guide what we should be doing with our patients. So this is a randomized open label controlled trial over 2 years, comparing the clinical outcomes in Crohn's disease patients who continue combination therapy. That's arm A, arm B, discontinuing infliximab, but continuing the immunosuppressant, and arm C, discontinuing the immunosuppressant, but continuing infliximab. So patients that were included in the study had to be in a steroid-free remission for at least 6 months and treated with infliximab and an immunosuppressant for at least 8 months. So in arm A, basically they continued the combination treatment, and if they had a relapse, then they would have to, they could intensify treatment to 10 mg per kg every 8 weeks. And at the end of the study, if they did not achieve remission, they were judged as a treatment failure. In arm B, patients could discontinue the infliximab, but continue their immunosuppressants at the same dose. And if they relapse, they could go back on the infliximab. And if there's no remission at 4 weeks, they go up to 10 mg per kg, then back to 5 mg per kg. Again, no remission at end of study, they're considered a treatment failure. And then the arm C group, they discontinue the immunosuppressant and continue the infliximab at the same dose. And if they relapse, they had intensification of the 10 mg per kg dosing. And then if there's no remission, an immunosuppressant was started. And at the end of study, if they did not achieve remission, they're considered a treatment failure. So just to give you baseline demographics, they were not very different amongst the groups overall. And they were all in an endoscopic remission, more or less. I mean, not endoscopic, I'm sorry, clinical remission. So the first co-primary endpoint was relapse rate over 2 years. And this was defined as a CDAI score greater than or equal to 250 at any visit between 150 and 250 with an increase in at least 70 points over 2 consecutive visits, 1 week apart with a CRP greater than 5 or a calprotectin greater than 250. Anytime they had new opening of perianal or enterocutaneous fistulas, that was considered relapse, or intra-abdominal abscess or perianal abscess, or if they had episodes of intestinal obstruction, that was confirmed on imaging. And the mean time spent in remission over 2 years was the second co-primary endpoint. So basically looked at the time computed from day 1 until relapse and added that time spent in the first and subsequent regained remission. They also looked at treatment failure, and they deemed treatment failure for patients who still were not in remission, even with treatment escalation for relapse, or if they had side effects or intra-abdominal abscess or intestinal obstruction. So this is the data. So for the first co-primary endpoint for relapse rate, at week 104, patients who were on combination therapy, their relapse rate was 14%. Patients who stopped Infliximab at week 104, their relapse rate was 36%. So these are the data you can tell your patients. Like if you stop your therapy, the likelihood of relapse is roughly 36%, by week 104, whatever. And then 10% a risk of relapse if you stop the immunosuppressant but continue the Infliximab. They looked at the second co-primary endpoint of mean time spent in remission over 2 years. So what's interesting is that, as I mentioned, if they relapse, they could go back on the Infliximab in arm B, and in arm C, they can go, So if they're in arm B, they stop the Infliximab, they restart it. And then in the arm C group, they can escalate on the Infliximab and add back the immunosuppressant. But what's interesting here is that when they restart therapies, they're able to regain clinical remission. So there's not really a significant difference in time spent in remission between the 3 groups. So this suggests that perhaps we can consider cyclical Infliximab infusions in patients with breaks. And I think this becomes relevant when we think about costs of therapies over time. So out of the 39 relapsers, 28 were retreated. So 7 were treatment failures. And of the treatment failures, there weren't significant differences between the arms of who were treatment failures. So the group that stopped the Infliximab, only 8% of those had treatment failure. And remission was achieved in 89% of patients. And then when you look at the Infliximab trough levels between baseline and end of study, even though patients in the arm B retreated group, you're able to regain the Infliximab trough levels, and there's not actually higher rates of anti-drug antibodies after retreating. And when they looked at predictors of relapse were being in arm B. So if they had higher, if they were stopping the Infliximab, if they were younger age, so less than 17 at time of diagnosis, or if they had high CRP or fecal calprotectin at the time of withdrawal of therapy, or if they had higher endoscopic scores. And then when they looked at multivariable analysis in the patients who stopped the Infliximab, they found that patients that had higher 6-TGN levels, this was protective for relapse. So in conclusion, Infliximab withdrawal, but not immunosuppressant withdrawal, was associated with higher relapse rates, so roughly 36%, than continuation of combination therapy. And there was a slightly reduced time in remission, but not by very much. And almost all patients who stopped Infliximab achieved remission when resuming treatment. So 89% of patients were able to achieve remission again. And overall failure rates were similar across groups. So younger age at onset and evidence of inflammation at baseline were associated with clinical relapse in all treatment groups, and low 6-TGN predicted clinical relapse in patients stopping Infliximab. And then if patients had development of stricture or baseline CRP elevations, that was predictive of treatment failure in all groups, and smoking was predictive of treatment failure in those stopping Infliximab. Okay, so next we're going to go on to safety. This is our last abstract we're going to present. This is looking at IBD in the older adult population. So we're seeing that as our patients are aging, it gets really confusing. Can we use the same drugs that we can use for younger patients? Do we need to really be worrying about infection risk or more adverse events in the elderly population? So this was a nice abstract that looked into whether we should be thinking also about comorbidities in these patients. Is that more important than the actual age itself? So we know that older adults are a heterogeneous population with differential risks. So the aims of this abstract were to assess comparative safety of anti-TNF agents, Vedolizumab and Ustekinumab in older adults with IBD, but accounting for the contribution of comorbidity. And when you look at other studies, when you look at risk of infection with biologic agents in older adults, you can see that the data are kind of all over the place, so it's unclear. So in this study, it was an observational cohort study using unidentifiable claims data from 2008 to 2019. And they were looking at US health insurance plan data. And basically what they did at the index date was look for first claim for anti-TNF, Vedolizumab, or Ustekinumab in patients that were over the age of 60. And patients had to have no prescriptions for the index treatment 180 days before continuous enrollment. And they looked during the follow-up period for infections, and they had to have no gaps in treatment during that time. So there had to be no periods of time where there was greater than 90 days between claims. And the end of follow-up was the date of last treatment plus 90 days or disenrollment. And so they had 3,693 patients with a mean age of 67 to 68. You can see that the Ustekinumab patients there were fewer because it was a newer drug at the time. There were so fewer prescriptions. And these were all the data were adjusted for the Charleston Comorbidity Index. So this index, basically a mild comorbidity index is like 1 to 2. Moderate 3 to 4, and greater than or equal to 5, you have more serious comorbidity. And their outcomes was time from treatment initiation to the infection-related hospitalizations. And then secondary outcome was time from treatment to any infection. And so when you look at baseline characteristics, they're overall pretty similar, except as I mentioned, Ustekinumab has a shorter follow-up period, fewer patients because it was approved later. And so here in the results, what you can see is compared with anti-TNF, the risk of infection-related hospitalization overall was not significantly different in Vedolizumab or Ustekinumab group. But when you incorporate the Charleston Comorbidity Index, so if they have mild comorbidity compared to anti-TNF therapies, you do see that Vedolizumab and Ustekinumab are protective. So you see slightly decreased risks of infection-related hospitalizations. So perhaps the comorbidity for these patients is more important than the age itself. And then when we look at the secondary outcome of any infection, and these are even not the serious infections that lead to hospitalizations, you're not really seeing any significant differences, even based on, even if you break down by the Charleston Comorbidity Index in terms of any infection between Vedolizumab, anti-TNF, and Ustekinumab. So in conclusion, older adults with greater comorbidity, Vedolizumab and Ustekinumab may confer a lower risk for infection-related hospitalizations compared to anti-TNF therapy. There were no major differences, though, in the rate of serious infections for older adults comparing anti-TNF, Vedolizumab, and Ustekinumab. And really, maybe comorbidity might be a more important index versus age itself when we're thinking about these therapies. So I've seen times when maybe the anti-TNF could have been more appropriate. And I think we are afraid of considering these therapies when we have other ones available. But if a patient seems to need the anti-TNF therapy and they don't have a lot of comorbidities, perhaps this could be something to consider. So with that, we are done. So we'll go ahead and we'll take any questions. Thank you.
Thank you so much to both of you. If you have questions, we have microphones set up around the room, and I don't know if you have access to the chat. Okay. Well, maybe I'll actually ask one question before we get started. So with this alphabet soups of the IBs and the MABs, I think for clinicians, what kinds of tools are being developed, if any, to help kind of stratify patients as they, kind of are being seen? Because it looks like you have different mechanisms of actions being targeted, obviously, right? So what strategies do you foresee in the future, or what kind of wish list would you have for a tool that would help you stratify your approach to patient care based on potentially either the mechanism that an individual patient may have contributing to their disease, or, again, what kind of devices or tools can you envision?
You're the best actually for this, because actually I think Dr. Dulai has developed some of these tools. So
there you go.
Go ahead.
Yeah, so I'm going to talk about it a bit in my talk that's going to be next here afterwards, but there are some tools that are coming available for you to use in practice. What I will say is that in the clinical trial landscape that's coming through, several of the companies started looking at companion diagnostics, where you'll actually have a serum-based test that you can check and say this patient is a high producer for this pathway and they would be appropriate for this drug. And so there's TLR sort of 4 inhibitors have a companion diagnostic platform. And so I think that is going to start to look more like oncology, where you have a test that just tells you this specific pathway is elevated. But we do have some immediate tools that we'll talk about when I sort of go through my talk that you can use at the bedside now for your consideration. So
great, thank you. Please go ahead.
Local GI and ask why. Quick question regarding when you start your patients on biologics, do you start them on regarding the immunogenicity? Do you start them on methotrexate and azathioprine? Is that something you do for a year? You never do? And how is that changing based on the data that you presented? That especially for Remicade, for Infliximab, that seems to be there's more benefit for to start those patients right off the bat with either thioprin or methotrexate, and wondering if that changed, is that going to change your practice at all or not?
Yeah, all right, I can take that question. So that's a great question. And I think that ultimately what we want to achieve is we want to make sure that for Infliximab, this is more specific to Infliximab, I think, because as we saw here, Ustekinumab, Vedolizumab levels don't seem to really change whether you're on combination therapy. Adalimumab, is a little bit not as, I don't have as much data for that, but I think that what we want to achieve is really higher trough levels, right? So if you do have access to drug levels, like regular drug levels, perhaps combination therapy may not be as necessary. I know that there's a camp of thought that we don't necessarily need combination therapy, but I think you do need to have regular access to drug levels. And that's hard to achieve, right? That's hard to get. So if you don't have that regular access, I really am a strong believer of combination therapy. I do believe that patients do better when you are on combination therapy. And what I do think these data highlight is, and I think as well, is that methotrexate also is a really nice therapy to consider for patients. I know that there's some toxicities and some adverse effects that have been reported for combination with thiopurines with Infliximab, but methotrexate seems to be a little bit, that adverse event profile seems to be a little bit lower, but it seems to achieve the same level, higher drug levels that we need. So I am routinely using that in my practice. And as far as the question of whether I de-escalate later on, that's a good question. There was another study that looked at, that I didn't mention, withdrawal of the immunosuppressants in patients. And I do think patients do have to demonstrate endoscopic clinical deep remission before considering any de-escalation. But, as we saw in the SPARE trial, 10% of patients can stop the immunosuppressants and don't have clinical relapse. So if you achieve that goal, perhaps you can de-escalate later. But I do think the combination therapy, if you don't have access to regular drug levels, are important in order to be able to really get that maximum effect you need.
So just to be clear, combination therapy, are you using that only for Infliximab, or are you using it for Vedolizumab and Ustekinumab?
So these data have changed the way I think too. I don't think you necessarily need it for Vedolizumab or Ustekinumab.
Okay.
Yeah, so that's what I was hoping to highlight here. I thought the data were really well presented this way. So before that was a little bit not clear, but this was a nice prospective study that highlighted that.
So moving forward, Vedolizumab and Ustekinumab, we're not supposed to be using any combination therapy based on this data. I'm just trying to frame that data into clinical practice, because in private practice, we do see, we want to optimize care for those patients, and we do have access to drug levels and antibody levels and all that. And just trying to understand, is this because for a lot of patients, they look at the anti-metabolites and they have, they look at it as, wow, this is an immunosuppressant. I don't want an immunosuppressant. So I think it would be nice to explain to the patient, well, if the insurance is only approving Infliximab, we have more data to support the use of anti-metabolites, at least I know sometimes we use it for a year, short period of time to decrease immunogenicity. So I'm just trying, since we have, IBD experts on the board, on the panel, just for us in private practice to better serve those patients. So let's say I get the patient today and I'm starting on Infliximab. What is your recommendations? And I do have access to serologies. What is your recommendation?
Yeah, so I also routinely use combination therapy for anti-TNF therapy, and I do not use combination therapy for Vedolizumab or Ustekinumab. And what you're getting at is the conceptual shared decision-making process that goes on with presenting to a patient the need to use a thiopurine along with the anti-TNF therapy. And I think one thing that's actually helped me is the risk immune test that's available, where you can actually predict whether they're going to get antibodies to anti-TNF therapy. And that's helped me convince some patients to actually use the anti-metabolite along with it up front. So that's a commercially available test through Prometheus that you can get. There was a really nice study done out of the UK, which shows that some patients are at a higher risk of getting antibodies to anti-TNF therapy, and those are the ones who benefit most from anti-metabolite therapy.
And that's a risk immune test?
Yeah.
Is that for, it gives you like a,
it gives you the
Trough and antibody? And all
no,
that?
it's upfront before you start therapy. It'll tell you whether this patient is at a high risk for developing antibodies to anti-TNF therapy. So even before you start. So that's helped me convince patients that they're going to benefit from this. But I do routinely present combination therapy as the appropriate first-line therapy for anti-TNF therapy.
And then if you end up on putting them on combination therapy, do you do combination therapy for a specified period of time or?
Yeah, I tell them that I would consider withdrawing the thiopurine if they've been in clinical and endoscopic remission for 1 year.
Yeah, I do the same thing. Yeah.
Yeah,
So we definitely both practice it. Combination therapy is really important, I think, for
Yeah.
infliximab.
Yeah, that's very helpful. Thank you.
We have one more question in the back and then we'll move on. Go ahead.
Yeah, so I have a patient who's failing VEGA, subtherapeutic levels.
For
with
ulcerative colitis?
For
or
ulcerative colitis,
Mhm.
Calpro is like 700 plus. And with the advent of the new orals, there's 2 out right now. So I'm going to use their brand name. So would you use Zeposia in this situation or Rinvoq? And this patient prefers to consider orals now as opposed to IVs or injectables. So how would you approach that patient?
Yeah, and so basically what you're getting at is they're having high clearance, not getting adequate concentrations of the Vedolizumab, and now you're thinking about switching.
Right.
We've had some success with escalating to every 4 weeks therapy, although the trial data would not sort of say that that's beneficial. But if you have a documented low trough, I might try to go to every 4 weeks first.
I actually presented that to the patient and she wants to switch to
She wants to switch,
orals,
yeah. So I think, so Rinvoq is only FDA approved after failing an anti-TNF therapy.
Right.
So if this patient has not failed an anti-TNF therapy, you cannot use the JAK inhibitor class. If they're anti-TNF naive, then I think going to Ozanimod is an appropriate next step. And that's the advantage of these small molecules, is that they are not limited by clearance. So you won't have the same issues come up that you're experiencing now for the Vedolizumab. Does the patient have anti-TNF exposure history?
No.
No. So I think it's really important.
Oh,
that-
actually, yes,
Okay,
Humira.
so then you have both options. So with anti-TNF exposure history, I would actually then think about Upadacitinib or Rinvoq. So I think
So
that
between the two, you'd go with
for
Rinvoq.
anti-TNF exposed, I would go with the JAK inhibitor because the data suggests that Ozanimod is not quite as efficacious in that class.
Yep, I agree. So anti-TNF failure, I typically will consider the JAK inhibition route afterwards.
Okay, one last question.
You didn't present anything on the safety. Is there any comparative between all the biologics in terms of safety profile long-term, comparing the anti-TNFs combination with the immunosuppressants versus the newer agents? Lastly, do you ever go the opposite direction when you've started Vedo or Ustekinumab? Do you go back to TNF, anti-TNFs if they fail?
Absolutely. So in terms of your second question, if someone has failed Vedolizumab, would you consider an anti-TNF therapy as a second-line therapy? Absolutely. I think that is a very plausible order of how to treat. I think that Vedolizumab actually is very nice first-line therapy for ulcerative colitis, and I think that it doesn't really limit. There haven't been studies suggesting that there's limitations of anti-TNF efficacy if it's used as a second-line therapy. Now, as far as the data for safety and comparing the different biologics for safety, I don't believe that there was any clear abstracts that would address the comparative safety of all the drugs. But there have been other, again, along the lines of the network meta-analyses we've seen that have addressed the safety of the various agents. I think that the safety data are a little bit relative to the patients in general. I think infection risks are a little bit higher that have been associated with anti-TNF therapies compared to the other Vedolizumab, Ustekinumab treatments. I don't know. Do you want to mention anything further on that safety?
Yeah, so for the newer therapies, the IL-23 class, so the IL-12, 23 and the IL-23 class looks very safe, looks to be on par with safety with Vedolizumab. The S1P class, for those of you who have used Ozanimod in practice, you have to do all this pre-screening with EKGs and eye exams and stuff. They presented some data at DDW that less than 1% of your patients actually would get screened out for some reason from that testing. So it's more of a bit more of a formality than it is an actual sort of safety concern. And there's really no new safety signals that were coming out for any of the newer therapies that we were starting to see. So I think that they're all relatively very safe. But definitely the IL-23 class looks to be like a safety profile similar to Vedolizumab.
Okay, great. Thank you both so much.